Immunologic tolerance. Immunologic tolerance is the phenomenon of unresponsiveness to an antigen induced by exposure to lymphocytes to that antigen. This is called immunologic tolerance. Self-tolerance refers to the lack of responsiveness to an individual's own antigens. It is called self-tolerance, and it underlies our ability to live in harmony with our cells and tissues, because the antigen receptors of lymphocytes are generated by somatic recombination of genes in a random fashion. Lymphocytes with receptors capable of recognizing self-antigens are generated constantly, and these cells have to be eliminated or inactivated as soon as they recognize self-antigens to prevent them from causing them causing harm. The mechanisms of self-tolerance can be broadly classified into two groups. One, central tolerance and peripheral tolerance. You can see this in figure 6.21. This is the figure 6.21 showing the mechanisms of immunologic tolerance. Nothing but self-tolerance to self-antigens. The principal mechanisms of central and peripheral self-tolerance in T and B cells are illustrated in this figure. APC indicates your antigen presenting cells, right? Now let's talk about each of these briefly. Central tolerance. In this process, immature self-reactive T and B lymphocyte clones that recognize self-antigens during their maturation in the central or regenerative lymph nodes. For example, the thymus for T cells and the bone marrow for B cells are killed or rendered harmless, that is, inactivated. Rendered harmless means inactivated. The mechanisms of central tolerance in T and B cells show some similarities and differences. The mechanisms of central tolerances in B and T cells show some similarities and differences. Now, in developing T cells, random somatic gene rearrangements generate diverse TCRs. Such antigen-independent TCR generation produces many lymphocytes that express high affinity receptors for self-antigens. When immature lymphocytes encounter the antigens in the thymus, many of the cells in developing T cells, random somatic gene rearrangements generate diverse TCRs. Such antigen-dependent TCR generation produces many lymphocytes that express high affinity receptors for self-antigens. When immature lymphocytes encounter the antigens in the thymus, many of the cells die by apoptosis. This process, called negative selection or deletion, is responsible for eliminating self-reactive lymphocytes. Self-reactive lymphocytes in the sense they will react against self tissues or self antigens. This process called negative selection or deletion is responsible for eliminating the self-reactive lymphocytes from the T cell pool. A wide variety of autologous protein antigens, including antigens that thought to be restricted to peripheral tissues, are processed and presented by thymic presenting cells in association with self MHC molecules and can therefore be recognized by potentially self-reactive T cells. A protein called AIRE, that is autoimmune regulator, stimulates expression of some peripheral tissue restricted self antigens in the thymus and is thus critical for deletion of immature T cells specific for these antigens. Mutations in the AIRE gene are the cause of an autoimmune polyendocrinopathy. You will see this uh, autoimmune polyendocrinopathy in chapter 24. In CD4 plus T cell lineage, some of the cells that see self antigen in in the CD4 T cell lineage, some of the cells that see self antigens in the thymus do not die but develop into regulatory T cells, which will be described later. When developing B cells strongly recognize self antigens in the bone marrow, many of the cells react to the machinery of antigen receptor gene rearrangement and begin to express new antigen receptors not specific for self antigens. This process is called receptor editing. It is estimated that a quarter to half of all B cells in the body may have undergone receptor editing during their maturation. If receptor editing does not occur, the self-reactive cells undergo apoptosis, thus purging potentially dangerous lymphocytes from the mature pool. Central tolerance, however, is imperfect. Not all self-antigens may be present in the thymus. And hence T cells bearing receptors for such autoantigens escape into the periphery, and hence the T cells bearing receptors for such autoantigens escape into the periphery. There is a similar slippage in the B cell system. Self-reactive lymphocytes that escape negative selection can inflict tissue injury unless they are deleted or muzzled in the peripheral tissues.